Okay, in the interest of time, let's get started. Thank you everyone for joining us for the Systems Biology Data Platform, Leveraging Accelerating Medicines Partnerships Research Opportunity Announcement webinar. A quick request as we go along, I'm sure you will have some questions. Please make sure to post your questions into the chat. Once we get to the end of the presentation, there will be a questions and answer session, and we will address any question that's posted in the chat in the order that they're posted. But please hold on to your questions until we get to the end. My name is Tony Kirilusha. I am a program leader at the NIH Office of Strategic Coordination and also the principal program manager for the SysBio initiative. We are joined by several other members of the NIH leadership team for SysBio. I will ask them to introduce themselves momentarily, and we will start with Dr. Robert Carter.
Good afternoon, Bob Carter, Deputy Director at NIMS and the lead for the sort of application to the Common Fund for this project. And I have to say, I'm really grateful for Tony for all this work.
Thank you, Bob. And Marianne Wu. We'll come back to Marianne. Katie Malora.
Hi, I'm Katie Malora, or Caitlin Malora. I'm the Strategic Planning, Evaluations, and Communications team member from the Common Fund side for the SysBio initiative.
And then Jana Eisenstein.
Hi, I'm Jana Eisenstein. I'm a Scientific Program Manager at NIMS.
Thank you, Jana. And Erna Petrich.
Hi, this is Erna Petrich. I will be an Other Transaction Agreements Officer for these awards.
And we're also joined by several working group members. I saw Stefania Delorso.
Hi. I work in the intramural program at NIMS, and I'm part of the working group for this project.
And then I also saw Shveta Ladva.
Hi, everyone. Good afternoon. I apologize that I think I appear multiple times in this meeting, but I'm Sveta Ladwa, and I'm the Chief of the Scientific Solutions Delivery Branch at the Information Technology and Application Center at NHLBI.
And Christine Swanson-Fisher.
Hi there, good afternoon. Chris Swanson-Fisher, Program Director in our Division of Neuroscience at NINDS, and also representing our Accelerating Medicines Partnership in Parkinson's disease. Great to see everybody today, and look forward to the discussion. Thank you.
Thank you, Christine. And Mette Peters.
Thank you. I'm Mette Peters. I'm a Senior Advisor in the Division of Neuroscience at the NIA. Thank you.
Thank you, Mette. The participant list is getting quite robust. Is there anyone else I missed? Please speak now or hold your peace until we're done with the presentation. And also, Jana, did you want to project slides, or did you want me to project the slides?
I can project those, and Marianne has joined if you want to introduce her.
Okay, let's get Marianne to introduce herself really quickly. Marianne, we can't hear you. I'm sorry. All right. Well, we have some technical difficulties, but Marianne is the program leader for the Venture program, which is the umbrella initiative, and SysBio is a program under the aegis of the Venture program. So Marianne will have a chance to answer questions after the presentation. But Jenna, let's go ahead and get started with the slides. So, and let's go ahead and go on to the next slide, please. So the agenda for this presentation is we will give a brief overview of what the Common Fund is and of the Venture program. Then we will spend some time discussing the SysBio research opportunity announcement. And anytime we have left over, we anticipate having at least 25 minutes, will be spent answering questions from the participants in the webinar. Next slide, please. And let's do one more. Okay, So the NIH Common Fund supports bold scientific programs that catalyze discovery across all biomedical and behavioral research. These programs are by design cross-cutting, and rather than focusing on one disease or organ system, they advance the mission of multiple NIH institutes and centers. Programs are designed so that each deliverable will spur subsequent biomedical advances that would not otherwise be possible without the initial strategic investment. Next slide, please, Jenna. And some features of the Common Fund programs and how they catalyze biomedical discovery. They make substantial but time-limited investments. What we mean by substantial is that a program is typically between tens to hundreds of millions of dollars, but they are limited to no more than 10 years. And some programs last only 4 or 5 years before they end. So each program will be at most 10 years, and the programs are goal-driven so that at the end of the program, we will be able to accelerate emerging science, remove research roadblocks, enhance the research workforce, and support high-risk, high-reward science. For example, for accelerating emerging science, the Human Microbiome Project expanded the field of microbiome research. And this was actually from a couple of years ago by taking advantage of recent advances in sequencing to characterize microbiomes of healthy adults on a scale that was never before possible. And our high-risk, high-reward research program supports exceptionally creative scientists pursuing highly innovative research with the potential for broad impact in biomedical, behavioral, and social sciences within the NIH mission. This includes 2 initiatives for early career investigators and 2 initiatives for established investigators. Next slide, please, Jenna. The Common Fund science and management are collaborative. First, we gathered diverse input from NIH leadership, staff, and the broad biomedical research community during strategic planning for each program. This includes identifying concepts that could potentially serve as a Common Fund program, as well as sharing and refining ideas once they are selected. We also assemble consortia of multidisciplinary innovative researchers who collaborate to tackle a shared ambitious goal. Because the programs are time-limited and goal-directed, everybody needs to make a collective effort, and diverse expertise is crucial for success. Finally, each program is managed in partnership with the institutes and centers. So each Common Fund program is run by a trans-NIH working group with leadership provided by several institutes or centers. ICD senior staff service program co-chairs providing leadership and guidance for our program. That's Dr. Carter for SysBio. And then institutes nominate members of their staff to serve on the program working group, as we have heard from a number of the members that represent multiple institutes that collaborated on making this research opportunity possible. Next slide, please, Jenna. Finally, a word about the Common Fund Venture Program, which is a new program. It was just established this year, and SysBio represents one of the 2 pilot projects that will encompass the launch of Venture Program. It is supposed to do amazing things with modest funding, which is a great and difficult goal. Venture Program will make Common Fund support available for short-term initiatives that embrace scientific risk. So as I mentioned, regular Common Fund programs are typically limited to 5 to 10 years. Venture initiatives are limited to no more than 3 years. So SysBio will be limited to 3 years duration. Ideas are prioritized by a venture board that's composed of 5 institute, center, and office directors who serve on staggered 3-year terms and represent a cross-section of institutes, both in terms of their relative budgets and in terms of the type of science that these institutes support. The criteria for venture program pilots are that they emphasize innovation and speed, and that the project should have potential for major impact and embrace scientific risk. Each project will be clearly defined with deliverables and go, no-go milestones, and will feature a flexible approach, as we'll discuss in a few slides, and will be managed in a nimble, streamlined, and flexible fashion. Next slide, please, Jenna. I will now hand this off to Dr. Carter to give us a brief overview of the scientific rationale for SysBio and the way it's going to promote science going forward. Bob, please.
Thanks, Tony. And I hope what you'll pick up on is that this project, the SysBio or systems biology data model, actually fits perfectly with the adjectives that Tony used to describe the new venture initiative. So I think, I presume most people on this call are aware that science has changed dramatically over the last 5 to 10 years, where with the emphasis now on understanding what's going on in human tissues at the cell and molecular level using multi-omic type approaches, so that both in normal tissues like the Human Cell Atlas or in disease tissues, there's been lots of data for different types of omics from the human tissues. The problem is a lot of these are stored on individual platforms that can't talk to each other. I do think what we found across these diseases is if you start doing comparisons, that you start to see similar cells popping up in different diseases that you really wouldn't have expected. You also see pathways that are common inside of cells, even though the cells may be different. They're, for example, inflammatory pathways inside the cells may be similar across different diseases. And I think we have to start thinking a little more broadly about diseases. Transcriptomics is great to understand, what's regulating RNA, but we need to understand the epigenetics and go one step upstream for that. Or we also know, say, in immunology now, that the immune system, the cells change their metabolism very dramatically with different activation states, and conversely are also activated in some cases by soluble metabolites. So we need to start integrating across these different omics, for example, to understand the interplay of transcriptional cell state and, say, mitochondrial function, if we're going to understand these systems. So that's that second bullet about to understand the disease and in humans. I think we have to be able to integrate across tissues to understand how pathways play out across tissues and how the pathways with different types of omics intersect. So that's the goal, that's sort of the larger goal with systems biology and data model. The idea here is we're going to bootstrap this. We want to get this off the ground where we can start doing these types of comparisons without having to go out and reach out and identify different data sets from different sources and go through the whole trouble of getting access to them and then downloading them, et cetera. And we're going to start using the data that's accumulated in the NIH's, it's a federated Accelerating Medicines Partnership, which includes, I think, I can't lose track, but at least 8 different projects, most of which are collecting some sort of tissue analysis with some sort of omics. And they're beautifully done data sets where there's a lot of attention paid to careful quality control, and actually in a lot of cases to understanding the humans from whom those tissues were obtained. So the idea behind SysBio is to start this work of being able to compare across diseases and across omics, but start with the Accelerating Medicines Project as a sort of test bed for how we're going to build a model that will then grow from that space. So as shown in blue, using the venture space funds, we can now hope to be able to explore mechanistic hypotheses across tissues, identify the shared mechanisms, and as I suggested, actually then link the patient phenotypes to what we're seeing going on in the tissues across diseases, no matter what the clinical diagnosis. Next slide. So the idea here is that we don't want to boil the ocean. We need to start small. We need to build a platform in 3 years with $5 million a year that can start this process. And a little bit small, but in the visual, in the image, it shows the different layers of different types of omics, including patient phenotype data. And then to how we can query those to look at different molecules in cells or RNA across different tissues. And then so where the SysBio comes in is we build that intermediate, that middle blot there, where we be able to query across the different platforms to be able to ask these questions in different tissues. And then moving to the right, it allows us to do the quantitation across the different data sets, and then from those to develop a systems biology data model that'll allow us to understand how disease functions in different tissues based on the same building blocks. So the impact of this will be a portal where one can query across the different data ecosystem. And the purpose of this is to understand the biology of disease. And also then, once we understand how patients with a single clinical diagnosis can have different pathways that are active. And in fact, patients with subsets of diseases and different diagnoses can actually have the same molecular patterns underneath them, so that we can both personalize treatment by understanding which mechanisms are active with which patient, no matter what the clinical diagnosis, and treat across diseases more effectively, what we call a molecular taxonomy of disease. Next slide. So this is called an Other Transactions Authority. It is not a grant. It's not a cooperative agreement. It's not a contract. But it is a nimble mechanism where we work with the applicant who, if after review, if you're scored well, we will work with the applicants to start. Tony, I think okay, you take it from here. And
I think this was where I was going to take over, although you're free to provide additional context. And,
no, you're right. And then I'll go back at slide 13.
I think what's going to happen is we're going to get to the end, and then if there are questions about the AMP program, we'll provide additional context.
Okay.
Based on the deck. All right. But actually, so thank you, Bob. And other transactions are in fact defined by what they're not, which has a couple of important implications to keep in mind. First of all, many public policies do not apply to OTs. So FAR and NIH grant policy do not apply. However, statutes still do apply to OTs. The other important distinction is that other transactions do not go through peer review, but they are required to undergo objective review. It does not follow the traditional NIH review process in a sense that they're not reviewed for the 5 statutory criteria that cooperative agreements and grants are reviewed for. Instead, the research opportunity announcement defines what the review criteria are. And so as you read the research opportunity announcement for SysBio, I encourage you to pay special attention to that section of the RAW, because it will explicitly tell you how the applications are going to be evaluated. And there will be a panel of experts that will be assembled to perform the objective review. The other important consideration is that other transactions are a very nimble mechanism. So they allow for relatively quick addition or subtraction of expertise tools, technologies, partnerships, and other resources that arise as the project develops. They support cutting-edge research, so changes in scope are anticipated and allowable under this mechanism, and there is no future commitment. So funding segments are in at most 12-month increments, and every additional funding segment is subject to meeting the go, no-go criteria and is treated essentially as a renewal. So that's the main difference from grants and cooperative agreements. Additionally, program can propose changes, although changes are subject to agreement by the awardee. And if the awardee is unable to agree on the changes, then certain actions can take place from there, up to and including discontinuation of the award. Jenna, next slide, please. So now let's actually talk about SysBio. That's what you all came here to hear about. And the research opportunity announcement was developed to establish a systems biology data platform that leverages data from multiple accelerating medicines partnership programs, which Dr. Carter just explained to you a few minutes ago. The full text of the research opportunity can be found at https://commonfund.nih.gov/sites/default/research-opportunity-announcement-OTA-2024-008 SysBio-updated-2024-0311_508.pdf. It can also be found by searching for OTA-24-008. So SysBio will provide a single access point for querying data across tissues and conditions studied by AMP. It will enable broad use of the data sets and knowledge generated by AMP, and it will consist at a minimum of a search portal and a linked cloud-based workspace. These requirements were separated into 4 key functional categories. Each of them is outlined in the raw, and we will discuss each one of them forthwith. Jenna, next slide, please. So function 1 is actually project management and coordination, which will be crucial for this effort, especially given the relatively compressed time scale that it must be accomplished on. The awardee will be expected to track milestones, ensure that the deliverables are timely, and ensure that progress reports and other program materials are submitted on time. The awardee will also need to organize SysBio meetings, which includes the kickoff meeting that's outlined in the raw, as well as working groups, both internally and with the AMP, the participating AMP programs. Those meetings are going to evaluate challenges and come up with decisions, so it will also be critical to develop a documentation and approvals tracking system to make sure that the outcomes of these meetings are disseminated to partners in a timely manner and correctly. The awardee will support external communications with both the scientific community and industry partners, because AMP is a public-private partnership, and we do expect at least some interest from industry. And finally, this is important. The awardee must present a plan to account for potential discontinuation of support before the 3-year period is up or at the end of the 3 years, as well as for potential transition of support of the SysBio platform to a new source. Next slide, Jenna, please. The second function, which is critical here, is data security. AMP programs contain a lot of participant contributed data. And so both the consent and privacy of the participants must be respected. And also, we must ensure that the data supplied by our industry partners are not accessed in an unauthorized manner. So the data security function can be broadly split into 2 parts. One is users. So users must be able to register themselves, establish their identity, and request access to controlled data. To that end, those requests for the data that they will be able to discover through the search functions of the portal need to be streamlined as much as possible while respecting the restrictions on the data and the consent forms that were signed by the participants that contributed the data. And the second part is the platform security. So in consultation with the NIH information security officers, it was established that, at least initially, the platform, which means the portal and the cloud workspace, need to be FISMA moderate compliant, which means both some upfront investment into the security posture and some ongoing evaluation and testing to ensure that the security standards are maintained. And in that category is also ensuring that SysBio or AMP data are not provided to a publicly available generative AI, such as ChatGPT or Google Bard or any other similar systems, because the security posture of those systems is very difficult to establish. And also, once the data are provided to those systems, it's impossible to know how it was integrated into the underlying model. So any large language models and similar technology developed for supporting SysBio must be developed and used exclusively in support of SysBio. Jenna, next slide, please. Well, function 3 is data harmonization, which will develop the underlying integrated data set and metadata catalog that will serve as at least the initial product of SysBio. So the awardee will have to develop an inventory of key AMP data sets and the associated metadata, and develop a solution that enables queries across AMP data sets. You will also need to develop tools to automate mapping between variables in the AMP data sets. And all of this is with the goal of producing a harmonized data set, or harmonized data that allows querying by a gene, molecule, or cell or disease type across the data sets from the different participating AMP programs. And next slide, Jenna. Finally, function 4 outlines the requirements for the actual SysBio platform. The platform will be owned by the NIH. So a part of this, establishing the platform, will be meeting the FISMA requirements outlined in the previous function and getting an authority to operate. The platform will include a portal that allows users to search the harmonized data by querying for specific genes, molecules, or cells, and also disease conditions. It will also develop and deploy a cloud-based workspace that allows users to work with AMP data that were discovered through the portal. And another requirement for the SysBio is a tool that allows users to select data sets for potential retrieval and the accompanying API to accomplish the retrieval based on a metadata query provided by the user. And finally, SysBio isn't going to live in isolation. There are many other rich data generating programs, both supported by NIH and conducted without NIH support, but still relevant. So it is expected that such data resources will be identified and that SysBio will be able to support integration and use of data from those resources. Example being the Common Fund data ecosystem, but it is by no means the only potential complementary data set that could be leveraged for SysBio. This concludes the primary technical specifications, but there are some other requirements in the research opportunity announcement that we wanted to draw your attention to. One is the requirement for including a plan for enhancing diverse perspectives, which is abbreviated PEDP. Here, diverse perspectives can refer to people who do the research, places where the research is done, as well as to who participates in the research as a part of the study population. The PEDP must include a description of defined activities and actionable strategies for the inclusion of diverse perspectives, a description of how the PEDP will bring advantages or unique capabilities to the project, milestones for evaluating the PEDP activity progress and success, and finally, the anticipated timeline for the proposed milestones. Please note that the PEDP is a part of the criteria that will be scored during the objective review, and you can find that when you look at the relevant section of the RAW. Next slide, please. The other requirement is that there must be a data sharing and management plan, which is an NIH-wide requirement. These requirements are largely covered in the body of the research opportunity itself. However, all applications, regardless of the amount of direct costs requested for any 1 year, must develop and maintain good documentation and manuals, training materials that enable their use. And any scientific data generated by the program, SysBio will not be generating primary data, but we will be generating secondary derived data and metadata indices. And it is expected that, to the extent possible, those should be made accessible to the scientific community as quickly as possible, either directly or through access to the search functions and other tools that they support. Jenna, next slide, please. There are also some requirements on PI eligibility and minimum effort. So more than one individual may be named as a PI, but one individual must be identified as the contact PI. The contact PI must be employed by or affiliated with the applicant organization. Now, we do not have citizenship or other requirements on the contact PI, other than they be employed or affiliated with the applicant organization. But there is a list of requirements on applicant organizations, which are eligible and not eligible. So please consult the RAW, and please address any inquiries about organizational eligibility to the contacts for SysBio. If a multiple principal investigator application is submitted, there must be an MPI leadership plan in the overall leadership plan. And this is separate from the PIs, but because of the complexity and project management requirements associated with SysBio, NIH expects that a separate key person be named as the program manager for the project, and the program manager must commit no less than 50% effort to SysBio. Next slide, please, Jenna. In terms of the timeline, the RAW was very recently released. It was released last week on Wednesday. On April 3rd, there will be letters of intent due, which we will discuss momentarily. Those are primarily for our information. They will help us gauge the level of interest and also start assembling the objective review panel by looking at potential conflict of interest, both institutional and personal. The letters of intent are strongly encouraged, but they're not binding, and they're not required to submit a full proposal. The full applications are due on May 15th by 5 p.m. local time of applicant organization. So if you are on the West Coast, they're due by 5 p.m. Pacific time, which would be 8 p.m. Eastern time. We will not accept late applications. This is very important to remember for this opportunity. And then between May 15th and June 12th, we will conduct the objective review. Based on the outcome of the objective review, we will begin negotiations with one or more top scoring applicants. Now, our intention is to make one award. However, this is another feature of the other transaction mechanism that I need to mention. It is possible that the best solution will require asking 2 or more applicants to join forces and provide a revised proposal that features some aspects of those applications. We will not share your proposal with any other applicants without first getting an okay from you, but a possible outcome of the negotiation is the request to submit a revised application with different partners. And we anticipate that the earliest start date for SysBio will be August 1st. And we also anticipate that it will be awarded no later than September 25th of this year. Next slide, please, Jenna. We already discussed the letter of intent. It is due by April 3rd, and we will really appreciate if you could provide the required information. It is not long. The requirements are outlined in the RAW. Next slide, please, Jenna. And applications are due, as I said, by 5 p.m. local time of applicant organization on May 15th. Late applications will not be accepted. And important to note that applications must be submitted electronically through Assist, and they must be sent by email as a single collated PDF to me at anthony.kirilusha@nih.gov and to the SysBio mailbox at sysbio@od.nih.gov. Next slide, please, Jenna. This concludes the formal part of the presentation. You can find the FAQ and additional information on the web at https://commonfund.nih.gov/venture/sysbio/FAQs. And you can contact the program team at sysbio@od.nih.gov. With this, I will conclude the prepared remarks, and let's move to the question and answer session. Jenna will read the questions that are posted in chat. If you would like to ask a question, also please feel free to raise your hand and we will identify you. Thank you.
Yes, and the first question is just a universal question that I'm sure others have from David Glazer. He wants to know if the information will be shared. And yes, we're recording this presentation. We will be sharing the slides and the recording, and our colleagues in the OD are going to be preparing an FAQ document that they'll be posting to their website.
Thank you, Jenna. Are there any other questions?
Yes, someone asks, what happens after the third year?
That is an excellent question. I will start that, and then I think I will actually ask Bob to weigh in on that a little bit as well. So the Common Fund Venture Program support for SysBio will end after 3 years. There's a possibility that depending on progress of the milestones and expenditure of funds, we will allow a no-cost extension to complete work that was already approved. But there will be no new common funding beyond year 3. However, it is clearly the intention of this program to establish a resource that will serve essentially as a go-to portal for reuse of accelerating medicines partnership data, and that there will be a next step. As to what that may look like in the context of the EMP program, I will let Bob speculate.
Thanks, Tony. So I think it's fair to say that some of the people on this group may be aware that there is an effort within the AMP program to do something similar, actually referred to as the systems biology of inflammation. That is not a done deal. There's support for the concept, but it is not funded. But the hope is that SysBio will start this process and that the AMP would pick it up and expand it. And that's a bit why I said that the scope of what we actually end up doing within this SysBio is what will be negotiated with an awardee. There must be a deliverable that we can point to, but the hope is that it will then be expanded by the collaboration with the partnership within AMP. I'll go one step further, and I think Tony is right. I'm sorry that the answer is vague, but there's no question in my mind that this type of activity of stop thinking within specific diseases, even within specific tissues, and I'm going to say it, even within specific institutes, but of working together to understand the data that across the institutes, across the tissues, across the conditions. And I think that will be an emphasis for Dr. Bertagnolli, the relatively new director for NIH. So I think the tea leaves are that this type of activity, even though it's going to require integration across NIH, which is difficult, I think will be a central part of what we try to do. And certainly Susan Gregorick in the Office of Scientific Data Strategy have been pushing for this. And I think it's, they're also developing a strategic plan that I think, I'm not going to quote anything, but I think is supportive of this concept. So I think this is a rapidly evolving landscape, but one that is anybody who's seen the data of different diseases and how similar they are across tissues and conditions, I think would be convinced that we have to do this to understand disease. So long-winded answer, but I don't think this is going to be turn off the spigot. I mean, absolutely not common fund, but I think we will have to find ways to keep this going.
Next question that came in, it says, I understand the platform developed under this program will be owned by NIH, but what about the ownership by the developers? And as a follow-on, is inclusion in COTS, commercial off-the-shelf components, allowed, encouraged, or discouraged?
So that's actually a very good and complex question. So let me give a couple of answers here. First, we do not have a position on the inclusion of the commercial off-the-shelf components, except as far as they affect usability of SysBio, because we want the resulting product to be accessible to as many researchers as possible and to have as low a barrier for access as possible. The goal for NIH-funded programs is to allow researchers free access to the data and resource, although obviously in the case of a cloud workspace, we do expect the users of the cloud workspace to support their own computation. That is not the expectation of SysBio, that we will pay for everyone's compute. But the use of off-the-shelf components is good as long as it doesn't hinder access. And also, it must factor into the plan for transferring support, potentially, of SysBio should it go to a new awardee, or should funding for the resource be discontinued. Bob, did you have anything to add on that?
No, I think that's right.
Okay, next question. How is this different from the current portals at Broad, the BD2K via the NIH OD Common Fund?
So I am not exactly sure which portal is being referred to, but I can tell you that BD2K is no longer an active Common Fund program.
The next question is asking about the federated data requirements. Is it the case that the data will be retrieved from the AMP sites on an as-need basis, depending on the research need?
That is also an outstanding question, and I think we are open to seeing how the applicants want to address that as we review the technical proposals.
Will non-funded applicants have access to the harmonized data platform toward the development of new grants in the field?
Absolutely. In fact, we hope that as many researchers as possible will use SysBio to drive forward their scientific use cases and enable their research, both basic and clinical.
The program structure and OT funding vehicle are specifically stated to support a high tolerance for risk. What do the program administrators see as the risky parts / key technical challenges of this program?
So I will start, and then I will also see if Dr. Carter has anything to add. There are a couple of components. One is the data in different AMP programs is heterogeneous. They can be combined, but one of the technical risks, and it goes to actually identifying what the data sets are and figuring out the most constructive approach to creating an integrated solution that allows a researcher to search across multiple data sets without having to look through each one individually. The other one is we are not sure right now what use cases are going to arise, and the goal of SysBio is ultimately to support use cases. So as scientists begin using the platform and start either breaking it in creative ways or coming up with new requirements, we anticipate that creating potentially additional milestones or additional challenges that will need to be resolved. And Dr. Carter, do you have anything to add there?
No, that's very good, Tony. I do think it is because of the nature of AMP, both that it will require working with the different AMP platforms, which is always a challenge. And then, as Tony said, harmonizing the data, I think, is a big challenge. And I think we're going to have to find tools. We can't do this manually, particularly since we're also talking about eventually being able to bring in outside data sets. So tools to harmonize metadata, either about the nature of the sample, how it was analyzed, dissociation, and about the human where it came from. I do think a lot of that language will end up being standardized, but we will need tools to do that. And then, the bottom line is that we're working across platforms. So doing comparisons is going to require getting rid of batch effects, and that's harder when you get different platforms and different tissues. But these, I think, they're challenges, and in each case, I know of examples where it's worked.
Thank you. Is there a portal or mechanism to collaborate or communicate with the various teams?
I'm sorry, and I don't think.
So Tony, the question is how to talk to the different teams. If you by that you mean the AMP teams, we on the NIH side are just starting to get the AMP partners together to talk through how this will work and plan. We'll have to have a plan at some stage by the time we're ready to negotiate with an awardee. So sometime by this summer, the NIH groups that control the different AMP platforms will have a process for the AMPs to talk to each other and with SysBio.
Is a pure cloud system expected, or can it include hybrid/institutional systems with cloud workspaces?
I am going to say that any solution that meets the basic security and implementation requirements in the raw will be considered. We do not have a mandate for one versus the other.
Are there any requirements to deploy workspaces that adhere to memory-safe programming language mandates?
There are none in the raw, and that is something that we would be happy to evaluate in the context of the technical proposal.
The next person says, I understand use of commercial generative AIs such as Gemini, ChatGPT are discouraged, but what about using the API for the proposed platform that will hold the data in the platform and not in the Google's cloud, for example?
Well, if I understand it correctly, the main issue is still that we do not want to feed SysBio or AMP data to BARD. So if the platform has its own machine learning functionality that it uses to either assist harmonizing data or assist querying, and it's either large language model generative or it's a discriminative AI, that is acceptable. But what we cannot have is essentially using that as a conduit to provide those data to ChatGPT or BARD and then have them end up outside of the controlled environment of the platform.
Can we submit an LOI for a particular function to give you all options to mix and match?
You may, however, each application should address all 4 functional areas that are outlined in the raw, because they will be scored as such.
And Tony, I might encourage anybody who thinks that they have a particular solution to a single function to find collaborators to come in together.
And is this just a visualization portal, or do you expect integrative analytic capability?
I'll actually, Bob, would you like to get started on that, and then I can weigh in?
So, again, this is one of those areas where we have a large vision, but want to make sure we get a deliverable. And I personally think that a platform that can query cells, proteins, molecules across the different data sets and return data for, there are tools out there now that can look at, identified particular cell types in data sets and even look at gene expression in identified cell types. So that to me is it's not a low-hanging fruit because of the difficulties I said with metadata and the fact that you're working across platforms. But that to me would be sort of a starting point. But you would definitely want more than that. And you would want, say, pathway type analyses where then you get individuals, where you get nodes and connections and, linked. So I think this is one of the crucial things in terms of we would like an applicant to come in with what they think is possible, starting with a minimal deliverable, if it could be like I described or otherwise, but with the capacity to work together with us to make sure that was most useful for the potential use cases.
And I will completely echo that and say that we do not want perfection to be the enemy of good here. We must have a deliverable, and we have at most 3 years to do it. But we're also very happy to review any technical responses to this effect in the body of the application.
Okay. Are there any predilections on the data type to anchor data models around? For example, use cases that start taxonomies from molecular views, scRNA-seq, bulk RNA-seq, methylome, et cetera, and append clinical phenotypes to them, or start from the clinical phenotypes instead?
So I would say,
let me take, So the discussions we've had with sort of our working group is that transcriptomics is probably the most common data type across the AMPs. And it'd be relatively easier to harmonize the metadata based around those types of files. So that's sort of a simple, not simple, because again, you're harmonizing, but that would be sort of to me, the first step. But you'd quickly want to be able to add things. And like I mentioned, ATAC-seq or epigenomics or metabolomics. I think getting the human data harmonized may be more of a challenge because there's a lot more descriptors, but there may be sort of a limited set where we start. But ultimately, like I said, you do want to understand how the activity of different pathways influences outcomes across conditions, presentation and outcomes. So start small with a limited number of data sets, with a limited metadata that you're going to harmonize, but with the capacity to build on that.
Thank you, Bob. I'll just add that we do not have a formal requirement.
Right,
of the
that's
data
right.
model, if that was a part of the question. But I support everything Dr. Carter said.
No,
about
that's right. We're not prescribing here. We're sort of giving a vision of what we preconceived come up with, but we're looking for technical proposals. And we're certainly open to new ideas.
Thank you. I don't see any other questions. We still have a couple minutes left if anyone wants to send those in.
Let's
So, yeah.
Okay, go ahead.
So you captured the ones in the chat as well as the Q&A, Jana?
Yes.
All right, over to you, Tony. How do you want to handle this?
I'd say let's give people another minute to see if there are any questions, and then if not, we can thank everybody for attending and declare victory a couple minutes early.
And we will use the transcript to get to develop the FAQs using these questions as the starting point.
Exactly.
Oh, Mete, good.
Yeah, I just want to expand a little bit on that last question concerning the data models. Although we're not prescribing the data models, we do want you to keep in mind that, we are looking for interoperability, whether that is metadata standards or technical standards that have the ability to integrate into a much larger data ecosystem. So, using standards that are already being used across a lot of these NIH-supported efforts will be advantageous.
And Susan, do you want to say anything?
Just to echo what Bob is saying in terms of how important this project is to realize the vision that Dr. Bertinelli has of creating a federated data ecosystem. This is really important for the AMP program, and it will hopefully build a foundation for future AMP work. And so I think we're taking this very seriously and we're all paying attention. And I just want to thank you, Bob, and everyone for bringing this together.
Thank you. And there is one additional question. Will the portal be transferred to CFDE at the end of year 3?
So I think that's a good question. And it will not be transferred to CFDE because the portal after 3 years will not be supported by the Common Fund. So it will not be supported by CFDE at that point. However, portal being interoperable with CFDE is one of the strongly desirable traits of it, especially at the end of the 3 years. And if there is continued support, then CFDE would certainly welcome any and all engagement from the portal and will be receptive to continuing that engagement going forward.
All right, that's it.
All right.
We're delighted that so many people could join on short notice, and the questions were excellent. So really appreciate it.
Thank you, everyone. I really appreciate everybody joining us. Please have a good afternoon.
Thanks to Jana.